Welcome back, everyone. It is the Water, Wetlands, and Watersheds seminar. It's fall 2021, November 17th, and today we are lucky enough to have Dr. Kate Hubbard with us. I'm going to hand the introduction honors to Christine. So Dr. Angelini, if you would, please.
Sure. Hi, everyone. Good afternoon. And it's with great pleasure that I introduce our speaker today, Dr. Kate Hubbard. As a little bit of background about Kate, she received her BA in biology from the New College of Florida and both her master's and PhDs in biological oceanography from the University of Washington. Kate is sort of an internationally and nationally known expert in harmful algae blooms and serves currently on the National HAB Committee and has done so since 2017. Currently, she is the director of Florida Fish and Wildlife Conservation Commission's Fish and Wildlife Research Institute, FWC-FWRI, and in that in that facility is the director of FWC Center for Red Tide Research. In this role as sort of a research scientist, she's working closely with a broad network of partners to sustain and advance comprehensive harmful algae bloom monitoring, which we're going to learn about today in her seminar. And in addition to mentoring students and junior scientists in this role, she's also serves as a co-investigator of the NSF NIEHS funded Woods Hole Center for Oceans and Human Health, which is based out of the Woods Hole Oceanographic Institute. And in this role, she's involved in both harmful algae bloom detection and forecasting projects that are across the United States. So it's exciting that she's going to tell us about what's going on here in Florida, but she definitely brings with her expertise that's, goes beyond Florida waters, and we're really thrilled to have her today. So Kate, welcome, and we look forward to hearing your talk.
Thank you so much for that introduction and the opportunity to speak today. And I'm going to share my screen.
Looks
okay.
Yep.
Okay, great. So I'm going to be talking about the toolbox that we use for observing and understanding harmful algal bloom dynamics. I wanted to just start out with some acknowledgements. Since sometimes I forget those, and I really wanted to thank a number of the different partners that are working within the FWC Center for Red Tide, FWRI HAB group, which includes a number of scientists, and the Woods Hole Center for Oceans and Human Health, and a number of the different colleagues and partners that we have tied to that, as well as numerous funding sources, including the National Science Foundation, NIH, NOAA, National Estuary Programs, and the state of Florida. And let's see if I can figure out how to advance my slides. And so I thought I'd start out with some conclusions, and then I'll dive into my presentation. So I work on HABs, as Christine mentioned, both in Florida and outside of Florida. Florida has numerous harmful algal bloom issues. These require innovative observations, and we need to be able to do those observations over event to interannual timescales. And that's really to help understand the drivers and potential mitigation strategies. This is something that's been evolving over time, where we're getting better and better at that. And sometimes we're re-examining some of the same questions. Sometimes our research drives us into looking at new questions. While the frequency and severity may vary across different types of HABs, coincidental and emerging events present unique challenges, opportunity for growth. We also have a number of different threats: human, wildlife, health, ecosystem, economic health. These all are challenging, and they're all related to harmful algal blooms on local to global scales. And so that's where this toolbox of HAB observations comes in, and it's really ever expanding in terms of what it entails. And really to do that, we need integrated systems and networks of partners. No one can do it alone. And the need for effective communication is really critical. And this is both within these networks of partners, but also to inform management and to help the public understand what might be going on and what the potential risks could be, and how to avoid those risks. So I'll try to provide some examples of these different types of observations, networks, tools that we're using right now to look at HABs, again, in different systems across the US. And then also I'll end with a little bit of insight into how that might be expanded outside of the US. So just to go over some of the different harmful algal bloom issues and syndromes that we have in the United States, there are a number of them. This is from the National HAB office that's operated out of Woods Hole, and they every year collate reports about all the different harmful algal blooms that occur. This is really focused on marine and estuarine systems. And if you look at Florida, you can see that there are a number of different issues that impact Florida. Some of those are shared with other regions of the US, and some of them are unique. This is a recent paper that we worked on to try to understand the overall trend in some of these events and syndromes that are especially relevant to human health issues. So each of the different types of HABs does produce a different type of human syndrome, and this is typically if with shellfish consumption or consumption of the toxins that are produced by these different HABs in mind. So Crynia brevis produces a suite of neurotoxic shellfish poisoning toxins. Alexandrium and pyridinium, they produce a suite of paralytic shellfish poisoning toxins, and then Pseudonychia produces amnesic shellfish poisoning toxins. And so these toxins are all neurotoxins. They have negative impacts on human health. They can impact you differently if you consume them in enough quantities. And so because of that, the US has developed robust monitoring systems, and these are in place to protect human health. And from these monitoring systems, we can look back and see over time if there have been an increase in these types of events across the US. And this is looking at the increase relative to the number of zones that have had events over time. And so when we look at Crynia brevis, which is shown in blue on the left, we see that there has really been no significant increase over time. The same with paralytic shellfish poisoning species. We have Pyridinium bahamense in Florida, but if we look across the US, there are other species, Alexandrium, that produce these toxins. So again, no significant increase in the US. One type of HAB where we see that there has been an increase is Pseudonychia, and I will spend some time talking about Pseudonychia towards the end of my talk. This is one where there have been several different areas that have seen emerging events within the past 10 years or so. And so that's something that is definitely on the radar where we can see that there is a significant increase. But some of these other HABs that we've observed for a long time, we're just simply not seeing those trends. And if we zoom in on Florida, just to give a sense of these different organisms and how they impact our systems, Crynia brevis blooms occur nearly annually, especially in Southwest Florida. Pyridinium bahamense blooms occur annually, and we've had new closure areas as of 2016. These blooms typically occur in estuarine areas that have longer residence times. And then for amnesic shellfish poisoning and Pseudonychia species, we've had 3 closures in Florida since 2013. So because we have all of these different HAB observations that are needed to inform management actions, including the closure of shellfish harvest areas, which in Florida is done by the Florida Department of Agriculture and Consumer Services, we have extensive monitoring programs in place. And this is a picture of our microscopy lab pre-COVID. Now everyone is spaced out 6 feet apart and spread out across the building. But in general, this lab is one of the hubs of our facility. There's a lot of activity going on, and we process anywhere from, sometimes we've gotten over 100 samples in a single day, and these samples come in from numerous partners. And we have a combination of both routine monitoring and event response. And this monitoring and event response, and really being the eyes in the water, it helps drive a lot of our research questions and efforts, and some of the needs to expand into new and different territories. But this is sort of the core set of observations. So when we get a sample in the door, one of the first things we do is check and see what's in it. Christine mentioned that I'm the director of the Center for Red Tide Research at FWC. This was established in 2020 and continues to expand, but it provides the backbone for enhancing existing partnerships that have been maintained for a long time, and also to try to develop new ones. We have more than 14 interdisciplinary projects that are designed to address monitoring, management, and research gaps. And these are informed by the Harmful Algal Bloom Task Force, which was reactivated roughly around the same time that the center was established. So these groups are all working in parallel to try to advance our knowledge of red tide. And a lot of the infrastructure that we use for red tide observations, forecasting, it provides that template for thinking about expanding to other harmful algal blooms and systems as well. And I know that there have been several presentations already in the seminar series on red tide. So I won't give a huge amount of background, but I did just want to provide some overview that blooms can last for anywhere from a few months to up to 30 months. That's the longest bloom that's been observed. And this is just sort of a history. We started monitoring at the state level more intensively in 1953. So our HAB monitoring database goes back, but we know that there was also a severe event in the late 1940s. And so just looking back over time at the duration of these events, you can see that a number of these events have occurred for up to a year or more. And that's important for thinking about the current event that we have that started back in December of 2020. We're now in the 12th month of that particular bloom, and I'll talk a little bit about that bloom today. But it's something where trying to understand the factors that cause variability from bloom to bloom is something that we're very interested in. And given that these blooms can not only occur for long periods of time, but also can occur across pretty extensive distances. This is just sort of an overview of some of the different areas that are impacted to show that they're variably impacted. We've had, as I said, nearly annual blooms in Southwest Florida. These are all the different observations of Perennia brevis, and this is only through 2019. But we have had different levels of events in terms of looking at the different areas. There's one presumed initiation zone, which is offshore, and that initiation zone occurs across much of the southern Big Bend region to the Sanibel area, so big swath of the Gulf of Mexico. It's thought that this area can potentially seed blooms in the Panhandle as well as in Southwest Florida. Southwest Florida blooms can also seed blooms in the Panhandle, and to the best of our knowledge, all the blooms that have occurred on the East Coast have been, they started in Southwest Florida and then were advected around the southern tip of Florida. So again, there's variability in the severity of impacts in these different regions. And we typically refer to a bloom once cells reach 100,000 cells per liter or more. So just to give some context on the bloom that we have right now, we put monthly summaries up on our Flickr page. And so starting back to December of 2020, this was actually one of the latest blooms that we have had on record. It was the latest one, but there were observations in mid-October of seabirds showing signs of brevetoxicosis. So signs that they had been ingesting brevetoxin that we weren't necessarily detecting at the time near shore. And then we can kind of fast forward to April, where we're still seeing the bloom more or less in Southwest Florida, but we've now seen an expansion up the coast, and that was due to anomalous currents that helped push the bloom further north. And then in June, we saw the bloom go into Tampa Bay and really take off there. We saw it happen a little bit earlier than that, but just for the sake of showing, it really intensified throughout the summer. It was a short-lived bloom in Tampa Bay compared to the overall bloom. It was, several months long. And then we've seen, into August, continued expansion up the coast. And this is showing October, where we also have seen the bloom all along the Big Bend region and into the Panhandle. So the bloom is transported quite a bit by currents, and then there are also local conditions that can be important for driving the intensity of these different events and at a more local scale. One of the things that we had this year was a drought early in the spring. And so that was something where some of our estuaries were anomalously salty, and Carnea brevis does have a pretty broad tolerance in terms of salinity, but there is a lower cutoff. And typically that can actually help keep it out of some of our upper estuarine waters. And unfortunately that year wasn't, this year that wasn't the case. There were other factors involved with the bloom, and I will, I can touch on those at the end if I have time, but for the sake of moving on, I'm just going to keep going. So this is something, a phenomena that starts offshore, is transported onshore at depth, and then we typically will see the bloom manifest in coastal waters anytime in the late summer to fall. Again, last year was a little different with the bloom first showing up in December. And then the bloom can last anywhere from a few months to, multiple years. And so because of that, we have tried to really enhance the number of routine surveys that we do, as well as in situ monitoring via gliders, buoys, remote sensing, other innovative technology that we can do. And so one platform that we've been using to help advance our routine monitoring is by partnering with NOAA AOML. And they've been conducting surveys for over 20 years in Southwest Florida with the focus on these are actually showing some of the recent cruises. These are from 2019. They were really focused on Monroe County and South. And with the severe red tide event that occurred in 2017 to 2019, they started to increase that spatial footprint. And it's something that we've been able to partner with them, Socorro, and others to help expand that footprint so that we have these surveys occurring every other month. I would love to see these go on into the future so that we have that same type of long-term coverage to be able to further address questions about drivers. So these are some images. These are the different sites that are monitored. And you can see that there are different lines that we've added on, and I hope you can see my arrow. But typically, again, these cruises focused on the more southern area, and then we've expanded now to include some more northern transects. With these surveys, we're able to use an imaging flow cytobot. We couple this to the flow through system on the ship, and what this does, it's an imaging flow cytometer. So it collects water samples autonomously as the ship is moving, and it processes approximately one sample every 20 minutes. And we're able to use artificial intelligence, image recognition software to pick out Carnea brevis. We have developed classifiers that are able to target Carnea brevis, but also a number of other species present in Florida's waters. And so this is just showing, also we use remote sensing to help track these events, and we use them also to inform our different field efforts. But so if we kind of put these three things together, we're looking at the pre-bloom conditions in 2019, sampling then in the peak of the bloom in November, and then when conditions started to improve by January of 2020. And you can see our cell counts overlaid on here, and then also the imaging flow cytobot showing again that we're seeing these things more or less show up in the same places. One of the neat things about the imaging flow cytobot is that we're able to get a sense of the status of the bloom in terms of physiology, and that's something that we're working on with research partners as part of the Center for Red Tide Research. But we're able, for example, to see when cells are becoming stressed or when they're actively dividing. And the first application of this tool was in 2018. And one of the things that was a little bit surprising when we applied it then was to see that cells were actively dividing even after the bloom had been going on for a long time and seemed to be dissipating. We were seeing that at the same time that we were seeing stress cells. One of the things that we did this year with the bloom in Tampa Bay, we were able to deploy it on our dock. We had counts of Carnea brevis that exceeded 10 million cells per liter. With this instrument in the water, we were able to see this growing population turn into a stressed population over the course of just a few weeks. So it's something that we're trying to introduce into a lot of our different programs to get a better sense of what's going on with physiology over the course of these blooms. And that's really important for, again, understanding mechanisms and drivers. We also are using moorings to help track changes in environmental parameters over time. These are some of the moorings that are maintained by the University of South Florida. The Center for Red Tide is supporting some of those efforts. There are other funding sources, especially through IUS, that are involved in funding those efforts as well. So again, it's a big partnership, but these provide really critical information about the conditions in the Gulf and how those relate to Carnea brevis physiology. One of the things that we get asked a lot is whether temperature can play an important role on bloom dynamics. And sometimes we'll hear that because it's warming and we're having summertime temperatures, the bloom's going to go away. Because it's winter and it's getting cold, the bloom's going to go away. And we can kind of look back to those long-term blooms where we know that blooms can survive, this particular organism can survive a suite of different conditions. It is particularly adept at using a number of different types of nutrients, but can also thrive under a wide range of salinity and temperature conditions. And these moorings really help us get a sense for those conditions in the Gulf and how they're changing. And one of the things that we know from a lot of the different observations that have occurred over time is that Carnea brevis is pretty much always present in the Gulf, not at bloom levels, but at background levels. And so again, that's important for understanding how this organism is maintained, but also what may seed blooms that come into the future. So another type of in situ observations that we're involved with include gliders. These are instruments. You can see one here that's being deployed. They move up and down on the water column. They have a number of different sensors on them. And so this is something that we've been working with both MOAT and USF to advance glider, routine glider deployments. And these are showing, this map shows the glider deployments for USF in 2020. So these occurred, over a course of different months and different seasons. And one of the things that we're looking for with the bloom initiation offshore is enhanced chlorophyll signal at depth. And so this just provides you with that general time series of conditions over the course of basically winter through spring to the fall. And we see that there is often a persistent chlorophyll feature at depth. The extent to which this represents red tide is still something that we're actively investigating. And there have been a few occurrences where we've been able to link it with red tide. There have also been numerous instances where we don't see red tide in this signal. So it's something where, again, this is a really great tool to be able to implement right now. Since we are in the 12th month of the bloom and we're still technically in that window where quote unquote new blooms could initiate, that's certainly been a concern with blooms that can over summer, where we may continue to see the previous year's bloom and then have a new bloom that gets transported in. That's something that we think probably happened in 2018. The extent to which that happened this year, we're not sure, but we do have both a MOAT glider and a USF glider out right now that were deployed within the past few weeks. And so again, we're trying to keep a better eye on what's going on offshore. And these gliders can be implemented with sensors, not just for looking at water quality, but also they can be used to help track telemetry animals, tagged animals. And so it can be something where there's a lot of potential for expanding the capacity of this type of monitoring. Something that we've also built into both the mooring systems as well as these glider systems is the implementation of nitrate sensors. And that's really important for getting a sense of nutrient conditions, how they're varying, not just throughout the water column, but over time. So again, as these networks continue to evolve and expand, we'll hopefully have more of this kind of routine monitoring data that helps us get a better sense of the potential drivers and the different factors that can impact severity from year to year. So this is showing Carnea brevis cell counts. These are produced through light microscopy. So that that team that I showed in the picture earlier on, and it just gives you a sense for, this is the 2017 to 2019 bloom. That bloom lasted for 16 months. That was obviously one of our longer, more severe blooms. The 2019 to 2020 bloom was actually, thankfully pretty well behaved. And then this shows as we descended into the present bloom that's still ongoing. And all of these different observations are provided to a number of different partners. And one of the important things I mentioned early on was that trying to convey this information in a number of different formats for managers, for the public, is something really critical to our work. So we do have an active Facebook page where we try to post a lot of information as well as bloom status updates. But we also have things like a daily sample map. And this is something that we started during the middle of the 2018 bloom, where previously we had issued status updates on the blooms twice a week. But now this is a GIS-based mapping system and it updates multiple times a day. So as samples are counted and entered into the database, basically that data is available pretty much as soon as we have it. And so that's something that, again, has been really valuable, and it gets a lot of attention and use from the public, which is something that we can track. And it's also something that we're trying to improve in terms of thinking about different types of layers that might be important to bring and build into that. We have the collaboration for the prediction of red tides. This is a short-term forecasting system that gives us a sense of where blooms are going to go, and it takes into account our cell count observations in order to produce those forecasts. We also work really closely with numerous remote sensing folks, and those include folks at USF and NOAA. And in particular, our data is combined with USF, different USF products, and that's something that can be downloaded in a Google Earth platform. And I'm going to show some of these. I showed some of those plots when I was talking about the 2019 bloom and the cruises. But also, again, our data is provided to them on a daily basis so that they can integrate them into that tool. The Gulf of Mexico harmful algal bloom forecast is issued by NOAA's NCOS, as well as now the Gulf of Mexico Coastal Ocean Observing System. And this is a forecasting system that has a number of different products. It integrates cell counts from not just us, but other states in the Gulf of Mexico, as well as other partners. And it provides forecasts of respiratory irritation and bloom intensification. Respiratory irritation is an unfortunate consequence when the toxins produced by Carnea brevis are aerosolized, typically during wave action. And when there are onshore winds, that respiratory irritation can become a lot worse as opposed to when there are offshore winds. There's also the beach conditions reporting system, which is operated by MOTE. This involves having trained sentinels at a number of different beaches, and that provides information about respiratory irritation and dead fish. So the Carnea brevis produces brevetoxins, and that can cause a number of different negative impacts on wildlife, marine wildlife. I mentioned the birds earlier, but we also see extensive fish kills, and we see marine mammal, sea turtle mortalities as well. And then a lot of this information about the blooms, as well as some of the public health risks, are collated on DEP's Protecting Florida Together website. And this is really meant to provide a comprehensive overview of water quality and algal bloom sampling. They also have a freshwater component. And so it's a really great resource. And again, these are different tools that are not just used by researchers, but they're also used by the public. And I'm going to give some examples of how we use these tools recently kind of together to conduct an event response. So this bloom moved up through the Big Bend region. This is showing some of the chlorophyll satellite imagery provided by USF. So we can see that there are these different patches, and the warmer colors indicate higher concentrations of red tide. Not all chlorophyll on the West Florida shelf is related to red tide, but typically once we know that there's a bloom ongoing, we know that a large part of that signal is in fact due to Cornea brevis. And so this is showing an evolution over time from the middle of October leading up to the beginning of November. And one thing that was going on, again, some of the different ways that we interact with the public, sometimes in ways that are perhaps less great than others. On the 6th, there was an Ironman race. The Florida Ironman race was planned for Panama City Beach, which is right here. And an Ironman race involves a long distance swim, long distance bike ride, as well as long distance run. And so all of those were intended to take place either in or along the Gulf of Mexico. And so there was a lot of concern leading up to the race about what the potential impacts of the red tide might be. And so we used a number of different tools to try to address those concerns. We worked very closely with the county Department of Health, a number of different partners, as well as the race organizers, so that they could ultimately make a decision about what the level of risk was for the different participants, and to be able to convey that information. So this is showing our daily sample map. So now you can see kind of a zoom in on what it looks like. And this was leading up to the race. And unfortunately, we were still seeing patchy bloom conditions the week of the race. We ramped up our sampling with partners in the area, and that included Department of Agriculture, but also FWC's law enforcement branch and our regional office up there. And so we were able to see that there were these patchy bloom conditions along the coast leading up to the race, as well as in St. Andrew Bay. And so we wanted to continue to do monitoring. So we kept looking at samples on a daily basis. We also were tracking fish kills in the area, because that, again, that can be a good indicator that there's an active bloom going on. And leading up to the bloom, we saw that, there had been some fish kills in the area when the bloom had been more intense, but by and large, we weren't seeing new reports. So that was a good sign. We were looking really closely at the trajectory of the bloom. So this is the forecast during that time period, leading kind of up to the race day. And this is one of the tools that I mentioned that's put out by USF that includes our cell count data. So both, and probably Bob Weisberg presented on this earlier in the seminar series, but basically our samples are input as well as drifters. And this is showing where over the course of 3.5 days, those different either samples or drifters are likely to end up. So we were looking really carefully at that to see if there might be introduction or advection of cells from another location and what might be going on with the observations that we were seeing. And again, this updates every day. So it's a really valuable tool for being able to get a sense of what might happen. We were fortunate to have some aerial imagery from a helicopter that went up with some FWC and county staff. And what they saw, so I should have started on my opening slide. That was a really good slide showing red tide, where you can see that sometimes it's really evident. In these cases, I've put question marks over some of the discolored areas that we saw from the aerial survey that was conducted. And these were indicative that there was likely patchy red tide in the area. And that's ultimately what we observed with cell counts as well. this was really important to look at for a few reasons. One, because the race was along shore, and two, because the satellite imagery does not do a good job of resolving things right up at the beach. So again, different types of tools. Our samples are collected, as I mentioned earlier, by a number of different partners and then examined in our St. Pete lab. However, just because the race was occurring on a Saturday, we also sent someone up to the Panhandle, and they were able to use this tool called the HAB scope. And it's a very affordable microscope that's coupled to an iPad, and basically a sample is added to the slide, and a 30-second video is recorded. And this also uses artificial intelligence to recognize swimming cells of Karenia brevis in the video. The video is uploaded to GCUS, and then a cell count is determined. And so we had somebody go up to the area, and the day before the race, on Friday, they were able to process a number of samples from the race area, and they were able to see that the counts in the area were actually below bloom levels, and so that was great. And this information, as well as our cell counts, feeds into the respiratory irritation forecast that I mentioned. Again, this is issued by NOAA and GCUS, and it provides 3-hour windows of the risk. This is based on both the cell counts that have been observed as well as wind conditions, and it's updated again as wind conditions change. So it's a really great tool, and we were able to use this combination of different tools to assess that conditions had improved. It got cloudy right before the race, so we weren't able to get good remote sensing imagery to kind of help further validate conditions. So just to kind of give a sense of how these different tools are used, and in an event response capacity, something that just happened very recently. Okay, so I mentioned also early on that Karenia brevis is one of the different types of HABs that we have in Florida. Florida has over 50 different species of HABs that occur, and some of the other ones that are really important for human health that I mentioned early on are Pseudonychia, which is shown here in green. One of the things that stands out about Pseudonychia is that it's nearly always present. And then Pyridinium bahamense, and we see that there's a more cyclical nature of these blooms. And so these blooms typically occur in the summer, and these different organisms likely all have different life cycle strategies that are playing a role in these bloom dynamics. And I'll go into a little bit more detail about Pyridinium and Pseudonychia, and also some other different HAB issues that we have in the Indian River Lagoon. So this is a paper that will be coming out soon in Frontiers. And so the Indian River Lagoon is on the east coast of Florida, and it's a system that's composed of multiple subbasins that are indicated here by the different colored dots. And we have done monitoring in that region for a long time. One of the things that has happened in the past 12 years or so is that we've seen a shift from what we would say to be our traditional HABs like Pyridinium to especially small celled HABs that are difficult to identify, like Oriumbra lagunensis. This is commonly referred to as brown tide. And so, one of the things that has happened with this change to these smaller celled algae is that we have seen some increased issues with water quality. And this was in 2016 in association with a brown tide event. And backing up just a little bit, some of the first issues with these small celled algae occurred in 2011, 2012, 2013. Ed Flips at the University of Florida, as well as partners at the St. Johns River Water Management District, have been involved in monitoring that system for a really long time. And so they were able to help document, a series of blooms that occurred then. And in 2012 and 2013, those were the first instances of these brown tide events. And we, of course, had a project focused on trying to look at developing and implementing tools for monitoring these cells. And I'll show you some of those tools in a minute. Brown tide's particularly difficult to identify. And when we had that particular project ongoing, we really didn't see any blooms. And this is showing dissolved oxygen over time. But I just wanted to kind of give the sense that this is when we were doing that development, that tool development. And we saw that cells increased dramatically in, basically right after our project ended, and then they ended up crashing. The bloom ended up crashing, and we saw this huge decrease in dissolved oxygen. And that resulted in these widespread fish kills that occurred, and they were really intense. They made national media, and it's something where we've seen persisting events since then, and I'm going to go into that a little bit. This is actually a picture of my mom, and she lives on the Banana River, and so she was one of our citizen scientists that was collecting samples before we started having more routine monitoring through our field lab in the area, which happened after this intense fish kill. But just to kind of mention that we have different ways that we engage citizens, and one of those is in our monitoring. And we have hundreds of active citizen partners that are sending in samples, and those include commercial fishermen, sometimes family members, but again, a wide array of people that as we see an event or a new situation arise, and we typically are expanding that monitoring. We use different strategies, but engaging citizens has been one that's been incredibly valuable. So diving in a little bit, this is what brown tide looks like under the microscope, and it's described as having an amorphous cell wall. It's really tricky to identify definitively. It's also small. It's considered a nanoplankton, so typically cells are on the order of 4 to 7 microns, and they really just look like small, small circles. They're hard to discriminate from other taxa. We have seen observations of similar looking cells in other systems in Florida. And fortunately, we have been able to use and develop new tools for specifically identifying Oriambra loganensis. And so we've been able to use those tools to rule out the presence of brown tide in other systems, but also to track brown tide in the Indian River Lagoon. And so this is just showing some of those tools. We call, when we are looking under the microscope, we call these Oriambra-like cells, just again, because we can't provide that definitive confirmation. These tools that we are using, one of them is an antibody-based flow cytometry method. This was developed in Chris Gobler's lab, and it was based on an antibody that was first developed in Tracy Villarreal's lab. So we've been able to adopt that method and use it in our group to continue monitoring. And that was one of the things that we tested in 2015 when there really wasn't anything going on, but then we've been able to use it in a number of different capacities. And then we've also been able to develop a quantitative PCR-based approach. And these both have a really good affinity relative to our microscopy-based identification. But again, as I mentioned, there are other cells that look like brown tide. And so having that confirmation has been really valuable. One thing that we would like to do in the future to try to move away from the antibody-based approach is to try to couple our gene-based assays with flow cytometry. So that's something that we're working on in the background. But having these tools has been really, again, beneficial. This is showing in the Banana River Oriambra loganensis versus Pseudonychia, which you can see is not quite as prolific a bloomer over this 6-year time period. And this is, of course, when we had our study to work on brown tide. And then this is when we started to do more intensive monitoring, weekly monitoring with the Fisheries Independent Monitoring Group at FWRI. And so that's been huge to be able to get a better sense of what's going on with these different events. And you can see that the brown tide event in 2016, it never really went away. Right now, we are still seeing hints of Oriumbra lagunensis now and again, but by and large, fingers crossed, things seem to be okay. One of the things that, if we look at, Pseudonychia, it kind of pops up and disappears in certain years. Pyridinium vehemence, I mentioned that cyclical nature of the blooms, where the blooms typically start and in the late spring, early summer, and then they wrap up in the late summer, early fall. And that's something that we see pretty consistently from year to year. And we know that this organism, Pyridinium vehemence, has a cyst that resides in the sediment, and those cysts will germinate when conditions are appropriate. There are a number of factors that we've looked at that can help mediate that germination. One is the amount of cold that they've experienced leading up to the time when conditions become more favorable for them to be able to grow in the water column. And we've also been able to look at these blooms to kind of get a better sense of what some of the drivers might be. And you'll see that with these Oriumbra lagunensis blooms, they occur, they start in different seasons, and then they can persist kind of like Carinea for these really long time periods. And so trying to get a better understanding of what might be influencing those bloom dynamics. So far, we know that there are a number of factors that may help this organism stick around in the system. For example, we know that they may have a resting stage, and the extent to which that's involved in helping to seed future blooms is still unknown. We know that they're able to use a number of different nutrient sources. And so that's something that's really important if you think about a bloom that's occurring for a long period of time. Being able to use recycled nutrients is something that can really be beneficial if they're able to take up different forms of nitrogen, that sort of thing. There seems to be a lack of grazing on these blooms, and so that can also be an important factor that can help growth and maintenance exceed loss. And then they also may be able to produce allelopathic chemicals, which will help minimize the presence of other species. So a number of factors might be involved in the persistence of brown tide in the IRL. And we have tried to do some modeling, and I'll just say, kind of going back to the prior slide, we weren't able to effectively model some of the drivers underlying Oriumbra lagunensis, likely because it is very complicated, but also it's tolerant of a broad suite of conditions and has behavior that may favor it. However, when we look at Pyridinium vehemence, we see that, in fact, this is showing pyridinium in blue, and that's the observed abundance. And then absence is shown along the 0 line here. And then we can look at that relative to the presence or absence of brown tide, which is shown in green. And one of the questions that we wanted to answer was whether temperature and salinity impact pyridinium cellular abundance in the Banana River, in addition to some of the factors that we know are important for germination, and also whether or not an Oriumbra lagunensis bloom could influence that. One of the things that we saw with that really persistent Oriumbra lagunensis bloom was that we had diminished pyridinium blooms in those years. And so when we, and this is a royal we, when we conducted some statistical analyses to try to address this, we were able to show with negative binomial regression that we could essentially approximate a model that could predict Carinea brevis that took into account temperature, salinity, as well as the presence of Oriumbra lagunensis blooms. And so this is something, and the other factor that was important for understanding what might happen over short time scales would be understanding, being able to input the number of pyridinium vehemence cells. So developing these sorts of models, again, that tie into things like for pyridinium, the models that can predict germination, they can help give us a better sense of some of the factors that may be involved in promoting these blooms. And then these types of time series are also really important in thinking about how some of these blooms might be mitigated in the future. And especially in the case of Oriumbra and Pyridinium, these blooms are occurring in areas, estuaries that have long residence times. Pyridinium occurs in Old Tampa Bay, which is another area that has long residence times, but also these Pyridinium blooms have impacted restoration efforts there. And this has been really important for thinking about the penetration of light when there are these persistent blooms. Then submerged aquatic vegetation, seagrasses, they may not be able to get the light that they need. They also may be directly competing with some of these small, these microplankton or nanoplankton for things like nutrients. And so these things all kind of play an important role in the status of the ecosystem. And we have had unprecedented numbers of manatee mortalities recently in the Indian River Lagoon, and understanding how these blooms play a role in that has been something that we have been working on, and thinking about also their role in diminishing seagrass coverage in the area. And so again, trying to think about potential mitigation strategies going forward really requires this intensive spatiotemporal coverage to get a better sense of what the drivers might be, and then also where they may not be really as helpful. One of the things that happened in mid-2020, we had actually a novel cyanobacterium show up. It was a nanoplankton, another nanoplankton, unfortunately, and it turned the water this beautiful shade of green. And you can see that showing up in multiple subbasins of the IRL and satellite data. This is provided by Tim Moore at FAU. And we were able to monitor this organism using microscopy. It did look different in live versus fixed samples. And so at first its presence was missed. Then once we kind of figured out what to look for, we were able to better track it, go back to some older samples, and we were able to see that this organism picked up during the summer. And this was in September, so it was still going strong then. And then as seasonal temperatures declined towards the end of the year, we saw a sharp decrease in the bloom across all of the different subbasins that the bloom had been in. And so this is something where we've continued to see this bloom, but not at these same levels that occurred in 2020, but we've been able to see the persistence over time. We have done, in terms of our toolbox for trying to look at this, an unprecedented bloom, something that our taxonomists who have tons of experience in terms of looking at these via microscopy, they weren't able to identify it. So we moved to looking at this using genetic methods. And first we were able to do just traditional Sanger sequencing to identify it using the 16S gene. That's a gene that's routinely used for taxonomy. Then we were able to design novel primers and work with existing universal primers to get an even longer fragment, because what we were getting was not aligning with anything that had been previously observed before at the genus level. So this likely represents a new genus. And then we were able to use nanopore sequencing to reconstruct the metagenome. And so now we have essentially the full genome of this organism, and that's something that we're using to get a better sense of its physiology. It does appear to have nitrogen fixation genes, and so that might be contributing to its persistence and its ability to, take up nitrogen from the atmosphere as well as the water. And it's also something that we're using to help get a better sense of its taxonomic affiliation. And so we are working on that more formally and hope to have a new paper coming out that will describe some of those results relatively soon. But again, sometimes the unexpected happens, and then having that toolbox that can be evolved is really critical. One of the things that we've done is developed qPCR primers for this new nanocyanobacterium so that we're able to go back through our samples that we've used for brown tide analysis in the past, and we'll be able to see if this thing really was a novel species that kind of picked up in 2020, or if it was something that had been around for a long time before. Okay, so switching gears to Pseudonychia, I mentioned that it's very prolific in Florida. We see it almost all the time. We've had the first closures in St. Joe Bay and the Panhandle, and this was due to the levels of toxins in shellfish. Those closures occurred in 2013, 2014, 2017, and they occurred in different seasons. But one of the things that we know is that there were particular species associated with those closures. We've also had some of the first closures in New England. Those started in 2016, have been pretty persistent since then. And then we've also, since 2015, had, well, since 2012 really had some investigations that have shown that the toxin produced by Pseudonychia, domoic acid, is found in wildlife all along the Alaska coast. And so I'm going to talk about some of these events in New England and in Alaska. One of the things that really makes Pseudonychia challenging is that there are over 57 species that have been documented. It seems like every time I turn around, there's a new species. This is something that I did my dissertation and postdoctoral work on. And what we know about this is that a lot of the species, but not all of them, appear to be able to produce toxin. And there are conditions that promote toxin production. So simply seeing a toxic species isn't enough to say that there's definitely toxin in the water. And that's a little bit different from Carnea brevis and Pyridinium bahamense, the other ones that produce neurotoxins that we're worried about. One of the things that we have seen in our studies of Pseudonychia is that these different species can, to some extent, act as tracers of water masses, but at the same time, they're responding to their surrounding environment. And so there's this, again, really close coupling of physics and physiology. And that's something that it seems to pop up in all of these different types of HABs. And so it's really critical.
Kate, I'm going to jump in for 1 second. The official, so some of the students may need to peel off. The official end of the seminar is 12:35, but it seems like the world of HAB problem is larger than larger than life. So if y'all have to run off to class, that's fine. But we'll try to save room for a question or two. If you can stay on, please do.
And I will try to wrap up. I'm almost at the end here. So this is just to show you the diversity of species. This was as of 2012, toxic species. This is just showing again that there's a lot more species that have been found, and there's been this really expansion in our knowledge of that. Molecular tools have been really critical for understanding these different events. And just to kind of give a sense of the emerging events in New England, one of the things that happened there, it seems like there was an introduction of a particularly toxic species, Pseudonychia australis, and that's shown here in these red dots amidst all of these other species. And this is something where we're using a genetic fingerprinting method, and we're able to track these species over time. And now we've seen that that bloom has reoccurred every year since that introduction. And we've been able to couple our physiology. And so we're able to grow these cells in the lab and see what temperatures they grow best at. And we're able to then look at that relative to what's going on in the ocean in terms of circulation. And so we had two different hypotheses. This is PhD work that was performed by Susie Clark at Woods Hole. And we were able to test these two hypotheses that looked at the potential introduction of this organism. And based on both temperature as well as the progression of the event, we were able to determine that this probably was introduced from a more northern infusion of water. And you can see that these are the temperatures that are favorable for growth based on this introduction pathway from the north. And that's something that we're continuing to follow up on. But the likely origin then was probably the UK. So somehow this species made it across the North Atlantic. So that's particularly interesting to think about relative to climate change. 2016 was also a year where there was anomalously warm conditions. And so trying to think about how these HAB events may be changing in the future is something that we're really interested in. Very quickly, shooting up to Alaska, this is a really fantastic paper that was led by Katherine A at NOAA, and it's just showing the pervasiveness of these different toxins in wildlife. Sometimes they can be the sentinels of these emerging HABs and emerging human health threats. And so that sparked a lot of interest in trying to monitor and map out Pseudonychia in the Arctic system. And one of the things that we found is that there are different areas where we find toxin. And this was kind of unexpected. These are really cold areas descending into subpolar areas, and then they're connected to our more temperate waters. And what we see is that there are different species associated with these different levels of toxin. So there are multiple species, and they do have different origins. Some are temperate, some are polar, some seem to be obligate Arctic species. One thing is that we think that this species may have an Atlantic origin. There's a lot of different water masses mixing together in the Arctic. And so it's another thing to just kind of keep in mind as conditions are changing. We may see that there are new and different pathways for these types of HABs to be introduced. And then this is my last slide, but trying to think about all of these different issues and the potential for these HABs to cause really devastating impacts. I was a part of an ocean shot, which is part of the Ocean Decade for the US, trying to envision what a global oceans and human health program might look like. And there are a number of different components to that, but thinking about the different technologies and sensors, that is really a critical hub that feeds into that, in addition to trying to understand a number of different factors like economic impacts, trying to involve the community, trying to build new capacity. And so again, trying to look at the connection between the ocean and human health, knowing that the ocean can negatively impact human health. And there are, again, a lot of different ways that we can look at that, and it's pervasive, not just in the US, but across the world. So with that, I will stop and say thank you. And thanks again for letting me go a little late, and for those of you that hung in.
Thanks, Kate. Let's give Dr. Hubbard a round of applause, real or virtual. I mean, it's hard to applaud such sort of a nightmarish story, but the science behind it is, of course, eye-opening. It's frightening, but also like a sort of amazing compilation from the molecular scale, or like genetic scale, all the way to remote sensing. So open it up for anyone who has a question, and I'll go check the YouTube real quick. Go ahead, Christine.
Sure. Yeah, sure. Kate, thanks so much. I wish I had heard this talk like 2 years ago. And anyways, it's just really comprehensive and really impressive. So thank you. And one of the questions I have is about sort of understanding like these blooms at these subsurface levels, and what you see as the frontier of, whether it's in the case of red tide or some of these other bloom events of understanding kind of the benthic pelagic coupling that may be going on, or simply subsurface dynamics. And with regards to the toolbox, what do you see as either urgent needs or oncoming emerging tools, whether that's really kind of expanding the sophistication of the imaging flow cytometer that you mentioned, or other tools, like how do we get better understanding of what's beneath the surface?
I think one of the things that's really important, I mentioned life cycle dynamics, and each of these different types of HABs has a different life cycle. And so whether or not that life cycle involves the sediment, whether it involves nutrients in the sediment, trying to understand persistence versus introduction into a system is really critical. And the role that the benthic coupling may have, whether or not that provides a reservoir of of cells that can seed future events, whether it provides nutrients that are able to help cells that are maintained in a system persist. There's definitely different potential there, and so trying to couple the types of sampling, like the glider observations, that sort of thing, with the cytobot observations. I think those are going to be really helpful for trying to address that more. And obviously looking at the sediment itself is really important too. And so that's something that some of the partnerships through our center are really focused on that.
Great, thanks. I'm curious, you talk about sort of the initiation process and sort of tracking blooms as these masses, and it's just I'm curious to know, like, are bloom initiation and onshore transport a once per year thing, and then you track them, or do you sometimes expect that there's bloom movement as well as a continued onshore sort of participation, right, from the offshore waters?
Probably to some extent both. And that's something where knowing that there's always vegetative cells present, there's always the potential. If you think about exponential reproduction, it doesn't take very long to have one cell turn into a bloom. And so whether that one cell is offshore or near shore, I think that there's potential, multiple mechanisms that can be involved in that development. But in terms of how things get concentrated through that movement from offshore to onshore, that's where the physics of the ocean become really, really critical.
Great, thank you. Any other questions? All right, well, let's give another round of applause. Thank you so much, Dr. Hubbard. We really appreciate your time today. And everyone else online, we'll have one more seminar. Let's see, is it next week or is it the following week?
Not next week, following week.
Okay, so we're off next Wednesday. So there'll be our last one. I believe it's Dr. Jason Evans. So thanks, everyone. Good to see you all. Take care of each other. Take care of yourselves. And we'll see you soon.